Hello students, hope you continue to stay safe. So now in this class we continue with what we started last time. That is, we saw the structure, function, development of the placenta. Now we look at the development of the fetal membranes, the amnion, chorion and their fate, and we will also look at the structure and development of the umbilical cord and learn how multiple or twin pregnancies can occur and what happens to the placenta and membranes in twin pregnancies. So these are the objectives. Now we recall that when implantation was occurring, that is when the blastocyst was implanting, right in the early week, that means by the end of the first week, remember we had the inner cell mass and the outer cell mass as shown in the figure, and in the inner cell mass a small cavity began to develop at around the day 8 within the cells of this inner cell mass. Now this is the future amniotic cavity as shown here, and this will gradually enlarge. So we'll see what happens to it. Now here you can see this cavity is lined on one side by cells of the amnion, which are known as amnioblasts, and on the other side you have the bilaminar germ disc forming. So the taller cells are the epiblast, and the lower cells in the slightly yellow are the simple cuboidal cells. Those are the hypoblasts. So that's the bilaminar germ disc. Now these amnioblasts that are lining here the amniotic cavity, as early as this end of first week, start producing amniotic fluid into this cavity, and you can see everything else is, implantation is occurring as normal. Now we are at the end of the second week of intrauterine life. Why? Because you can see the implantation is complete and the uterine epithelium has grown back. So what is the structure we have seen? We've already seen this in previous classes. So this is the amniotic cavity that started developing in the inner cell mass. That was the original blastocyst cavity, which became the primary and then the secondary yolk sac. And this is the chorionic cavity, or the extra embryonic coelom, that developed by splitting up of the extra embryonic mesoderm. That is the brown tissue that is shown here. Now outside, that is the, remember, rest of the outer cell mass, so the cyto and the syncytial trophoblasts and all that. So we are not concerned with that. And this is the connecting stalk where this extraembryonic mesoderm did not split. Now note, this star-shaped cavity, that is the chorionic cavity, has enlarged so much and is lined by the chorion. Chorion means these three layers together: extraembryonic mesoderm, the cytotrophoblast and the syncytial trophoblast. And the connecting stalk where the mesoderm did not split will be the future umbilical cord. So let's see what happens further. Now we are going to find out what's happening, or we are going to learn what's happening to the amniotic cavity as all these developments are proceeding. Now in these figures, if you trace from A to B, C and D, what is happening to the embryonic disc? It is undergoing craniocaudal folding, because this is a sagittal section passing through the germ disc. Okay, and you know why craniocaudal folding occurs: due to the development of the brain, which is rapid. Now we follow, as a result of the folding, follow what's happening to the amniotic cavity, which is here, and follow these two arrows shown from A and B onwards. So as this embryo is folding, now AC represents the amniotic cavity. Note, it is enlarging and it is also folding or coming ventrally like that. And note in the figure C how it has expanded more and it is coming also ventrally. At the same time, you see what's happening to the yolk sac. We have already learned this. It is getting absorbed partially into the embryo and will form the future gut, and partially the yolk sac remains outside and is connected temporarily to the absorbed part by the yolk stalk. So this connection is called the yolk stalk. Now in this figure D, you see how much the embryo has folded and how much the amniotic cavity filled with amniotic fluid has enlarged, so that, now remember, these were amnioblasts, right, or amnion, we say. The membrane is called amnion. So this amnion now is come so ventral that it will actually come to line the future connecting stalk. So let's see further. So this is again a picture of the folding embryo and the yolk sac getting absorbed and the enlarging amniotic cavity. So follow this white cavity, and here you can see how much it has enlarged. And this blue is the original chorionic cavity lined by chorion or extraembryonic coelom. Now, from the 3rd to 8th week onwards, which is a major period of also embryogenesis, look what's happening to the amniotic cavity. It enlarges and there is increased secretion of amniotic fluid, which fills the whole cavity. Now this is the white cavity that we are talking of, okay, as the embryo folds. Now, as a result of this whole amniotic cavity surrounds the embryo and even lines the future connecting stalk or the future umbilical cord. So see, it gets lined by the amnion because of the way the embryo has folded. And now see what happens to this blue. Original blue was the chorionic cavity. See how large it was. But now, because of this enlarging amniotic cavity, the chorionic cavity is getting obliterated or disappearing. How does it disappear? Because this amnion, that is this lining here in the previous picture, will fuse with this chorion. See, the amnion has fused with the original chorion. So you get a fused membrane known as the amniochorion. And now this growing fetus, you can see, can swim around in this cavity, that is the amniotic cavity, which is full of fluid. So it has plenty of space to grow and exercise within. Like a fish, it will swim within this cavity. Is this understood here? And note the yolk sac again, part of it absorbed to form the future gut tube, and the rest will degenerate and it remains temporarily connected by the yolk stalk, which is within the connecting stalk. Okay. Now this is where we are again. So here this is in relation to the whole uterus and walls. So again, note, we start from inside. What is this cavity? This is the enlarging amniotic cavity. That's the connecting stalk. This is the original chorionic cavity. So what will happen? This amnion will fuse with the chorion. See, this is the chorion, and become fused amniochorion. Now note what's happened to the placenta. At the original site of implantation, the chorionic villi have grown, and so that is the fetal side of the placenta. The maternal side of the placenta is formed by decidua basalis. Okay. Now remember, chorionic villi had grown all around the chorion, but they don't grow that much except at the site of the placenta. So these shorter chorionic villi will ultimately disappear. Okay. So note here the enlarging amniotic cavity, the chorionic cavity eventually disappearing as amniochorion fuse. The chorionic villi are disappearing at the abembryonic pole. Abembryonic means all this around here, and that part of the chorion will become smooth. Now the placenta is only present at the embryonic pole, and the yolk sac is again seen degenerating. And note now the different parts of endometrium. So what do we mean by these terms? So decidua basalis, you already know, is the maternal side of the placenta. It's the endometrium that is known as decidua basalis, where all the lacunae, glands, etc. are maximally distended. Decidual reaction is occurring. Now remember, this entire embryo with its cavities, etc., had got implanted within the endometrium, right? So there is endometrium still covering the whole structure, and that endometrium is known as decidua capsularis. So it's still within the endometrium. And then note this all the other parts of the uterine wall, there is also endometrium, and that is known as decidua parietalis. Parietalis means wall. Okay, this is all endometrium here. So do you understand the difference between decidua basalis, decidua capsularis, and decidua parietalis? So we'll see what happens to these finally. Now here you note that again this is the same kind of schematic picture. So again note, this is the amniotic cavity, connecting stalk, chorionic cavity which is reducing because amnion and chorion will fuse. That's the site of the placenta. Okay, and then note, covering this whole thing is decidua capsularis, this is decidua parietalis, and this is known as decidua basalis as shown in the figure. Now, what are the parts of the chorion known? We've already seen this, that the part where the chorionic villi develop, that is known as chorion frondosum, like branching, and that's the site of the placenta. And the rest of the chorion where the villi are disappearing is known as chorion levae. Okay, we'll see in the next picture. So now in this, from this to this, what is the difference? Here you can see the amniotic cavity is enlarged so much and fused with the chorion. Okay, so hope you have understood how all this is happening and where we are. Now this decidua capsularis that is covering here starts becoming so thin that gradually it will disappear. And then see in this final picture what happens is that the fused amniochorion, chorion is outside, amnion is inside, fused amniochorion now is, or we also say this is chorion levae, right? The smooth chorion levae. So the uterine cavity is totally now obliterated, that means filled up by fusion of this chorion levae, or we can say amniochorion, and the decidua parietalis. So it's like as if now the whole embryo and fetus has, surrounded by its membranes, has come inside the main uterine cavity, which is filled up by the membranes and the growing fetus. I hope this is clear. So that's where we are. See, now this is where the placenta, that is the chorionic villi, and the decidua basalis will be above that. So this is chorion frondosum. Rest of the chorion, the chorionic villi have disappeared, and then it's called chorion levae. And inside there, so this is the fused amniochorion, we can say the embryo. See, you can see partly the little embryo growing inside there. Okay. Now let's learn something about why this amniotic fluid is secreted in the amniotic cavity. So it's a clear watery fluid produced by the amniotic cells, and some part comes from by diffusion from maternal blood. Usually this amount is very specific, so it's around little, 30 ml at 10 weeks, 450 at 20, but at towards term 37 to 38 weeks it should be at least 1 liter, 800 to 1000 ml. If it is less than 800, then it's not enough. Okay, we'll see why it's not enough. Now see, this fetus is growing and this is the fluid in the amniotic cavity. Now from 5th month onwards, because all the systems have developed, the fetus starts almost drinking, actually swallowing 400 ml a day, which is recycled. That means it comes out as fetal urine. It's not really urine because it doesn't contain waste products, and you know, the kidneys start functioning, primitive kidneys, so they secrete, and this is all recirculating, and this fluid is replaced every 3 hours. So it's like a constant amount depending on the age of the fetus. Now why is this so important? Because again it contributes to nutrition. There are substances, nutritive substances, which when the baby is drinking can, you know, get the nutrition. Then very important is it's a big shock absorber and a protective cushion. So imagine any bump to the uterus or shock hit to the uterus, but the fluid is a great shock absorber, just like our brain is surrounded by the cerebrospinal fluid, which is very protective. Thirdly, importantly, it allows fetal movements. The fetus muscles, bones the muscles need to be exercised. So because he's going to spend 9 months in it, so it allows fetal movements, and because the fluid is there between the membrane and the fetal skin, it prevents sticking of the embryo to the fetal membranes, to the amnion, which can affect its growth, as we shall see. So during parturition, means labor, it forms, because it's fluid. Now the baby is getting ready to be born, its head is, in a normal delivery the head is usually low, head comes out first, so below the head a bag of waters forms. Okay, and when the time is up, when labor, at the onset of labor, this bag of water or this membrane will rupture. So it's rupture of amniochorion actually, and then the leaking of the fluid, which is a sign that labor is beginning. So it's very important for the onset of labor also. But imagine if it ruptures prematurely, before the baby is fully grown inside, fetus is grown, then it can cause early labor. Okay, suppose there is trauma to the membrane tears off somewhere or something, then it can rupture early and cause problems. Okay. Now, so we have seen how precise the amount is depending on the age of the fetus. So sometimes there is excess for that age. So this is known as polyhydramnios or simply hydramnios, which means excessive amount of amniotic fluid. And when can we say it's excessive? When it exceeds or it's more than 1.5 to 2 liters or 2000 ml. And as you can see, there'll be lots of fluid here. Now, you know that a pregnant mother is undergoing repeated routine antenatal checkups, so she will undergo ultrasounds, and this can be detected during pregnancy. And some of these causes, imagine so many causes, 35% are still unknown why some women will have excess amniotic fluid, but it's definitely associated with maternal diabetes, also gestational diabetes, and or these are maternal causes. But for the fetus, you know that we've learned how the fetus has swallows it and it circulates within the fetus also. But if there are congenital malformations, you can imagine any gut anomalies, so the fetus cannot swallow, or CNS abnormalities, brain is not developing, GIT, esophageal atresia, etc. So then the amount will keep on collecting, it won't circulate normally, okay, and will become more. And that also can affect the fetal growth. Now, when the amount is too little, it's called oligohydramnios, and this is usually considered oligo when it's less than 400 ml for that age. Now, these causes, this is rarer, but then you suspect some renal anomalies. So obviously, you have to check by ultrasound for renal anomalies in the fetus, or there may be leaking somewhere, slow leaking of amniotic fluid, and this can affect fetal growth because if there is less, you can see how the fetus has no place to move. Its skin will be touching or get stuck to the membranes, and can cause limb defects or any other congenital defects. So these are both are associated with increased incidence of congenital defects. So to make this amniotic fluid can also be used as an antenatal diagnostic test to diagnose whether there are any congenital or genetic abnormalities. See, most gross anomalies you can detect with ultrasound, but this is like a microscopic examination of amniotic fluid. So how to collect it? So you can see here it is done under ultrasound guidance. This is an ultrasound probe, and then a syringe is inserted, needle is inserted. Either it can be inserted through the abdomen, so that's transabdominal or transvaginal, and basically you're accessing the amniotic fluid in the amniotic cavity, and a small amount, usually 20 to 30 ml, is withdrawn for examination of fetal cells, because fetal cells do rub off and are shed off and are collecting in the amniotic fluid. This is the safe period is around 16 to 17 weeks of intrauterine life, because there should be enough amniotic fluid. Also, the fetus should not be injured during the process. Usually the risk is minimal. So it's a useful prenatal diagnostic test, especially to detect genetic or chromosomal anomalies. Another prenatal diagnostic test is known as CVS, or chorionic villus sampling. We will learn about this in a separate class. So this is also known as transcervical chorionic villus sampling, means through the vagina, cervix, biopsy needle is passed, and now you are taking a biopsy of the chorionic villi, basically of the placenta, fetal side of the placenta, or the villi here. Again, it's fetal tissue, so it can be examined for genetic chromosomal anomalies, because you get fetal cells, and both these are done under ultrasound guidance. So here is a same thing. Now you can understand how once you get these cells, this fluid, you can centrifuge, the cells will settle down, and then you can do karyotyping and whatever else you want to do, genetic analysis, etc. You can do cell culture, and then use all these information can be obtained. Now see what happens if the fetal membrane, that means the amniotic membrane, suppose it sticks to the part of a growing fetus. It will hamper growth, and sometimes it may even cut off a limb like a sharp knife. You might think, how can a membrane do that? But if they can form like bands around the fetal limbs, causing these narrow areas, or here you can see it's almost like a cut off finger, or here the thing has got the leg itself has got amputated, so there might appear ring constrictions or amputations. So hence the importance of amniotic fluid. There should be enough so that no part of the fetus sticks to the amniotic membrane. Now let's look at how the umbilical cord develops. So what is primitive umbilical cord, what is mature umbilical cord, and what are its contents? So in the figures we have seen how the connecting stalk forms, because the extraembryonic membrane, mesoderm does not split up in the region of the connecting stalk. This is going to form the future umbilical cord. Now again we have seen what's happening here. As the amniotic cavity enlarges, the amnion, see the white, follow the white, this is amnion, right? Membrane that comes to surround the connecting stalk, which has moved ventrally. Now this is the primitive umbilical cord. As embryo folds, you can see the yolk sac is partly absorbed, partly remains outside, okay, and the yolk stalk connects the two parts temporarily. So the yolk stalk is a content of the primitive umbilical cord, or the connecting stalk. You can see here, okay. Also note that this allantoic diverticulum had also, from the yolk sac had gone into the connecting stalk mesoderm. So temporarily again the allantoic diverticulum is also a part of the primitive umbilical cord. Let's see what else is present here. So now here note at the junction of the embryo and the whole connecting stalk, here this area is known as the primitive umbilical ring. Basically it's a junction of the skin, and where all these things are coming in and out. So it's at the junction of where, now this is amnion, see it surrounds the whole connecting stalk as we said. So it's at the junction of the amnion and the embryonic skin or embryonic ectoderm at around the 5th month of intrauterine life. Now this is the whole primitive umbilical cord. Let's see what it consists of. So this is original connecting stalk, which is what? Mesoderm. So look at that orangish tissue there, that is extraembryonic somatic mesoderm, containing what? Containing the, so see here this is the mesoderm, containing the allantoic diverticulum. It's not shown in figure A, but here it is, and also the allantoic diverticulum as seen here, as also the umbilical vessels. Can you see these umbilical vessels? Yes, that is, you can see the allantoic diverticulum. See that little diverticulum in the middle here, that little pinkish tissue, and these are the umbilical vessels that have developed. What are these umbilical vessels? There are 2 umbilical arteries and 1 umbilical vein. What else does the primitive umbilical cord contain? So you can see the yolk stalk, which is connecting the outer part of yolk sac and part which is got absorbed inside, or here this you can see the yolk stalk here, right? Remember, this is a temporary content. Now because this yolk sac is there, it has its own blood vessels. So you can see some small blood vessels here. These are the vitelline vessels or the yolk sac blood vessels, okay. So they are also a temporary content. And finally the third thing is the canal. Canal means just a space. See this canal all around, little bit here, which is just a canal connecting the chorionic cavity or extraembryonic coelom with the intraembryonic cavity, okay. So that's just a canal. So to repeat, connecting stalk with all these mesoderm, allantoa and umbilical vessels, the yolk stalk with the vitelline vessels, and the canal connecting the two intraembryonic with the extraembryonic cavity. Is this okay? Now the development continues. Remember, there in the 5th week it had all these primitive contents. Now as 6th week of development proceeds, something else comes from the embryo outside into this connecting stalk. And what is this? So between 6th and 10th week of intrauterine life, an additional content of the primitive umbilical cord is the loops of developing intestine, specifically mid-gut loops. And you can understand this why here, because this absorbed part of yolk sac, this is foregut, future midgut and hindgut. Now imagine if this gut tube is developing, your intestines are so long, right? So they don't have space in the growing embryo because the body wall is small. Hence, as they grow, they come out into the, as you can see here, into the connecting stalk mesoderm or the primitive umbilical cord, and that is the yolk, rest of the yolk stalk attached to it anyway, because this was original yolk sac only, right? Now this herniation of the intestinal loops into the primitive umbilical cord or into the connecting stalk mesoderm, this is known as physiological hernia because it occurs normally. And when does it occur? Between 6th to 10th week. So 6th week the loops start coming out, and by 10th week they will start going in back again. So loops will start returning to the embryo in this period, 10 to 12th week. Why? Because then by the time the whole embryo has grown so much, there is more space inside the embryo, so the loops will go back inside. Okay. Now after the 12th week, what happens is the changes. The loops of intestine have gone into the embryonic abdominal cavity. The yolk stalk and unabsorbed part of yolk sac, which was outside, they have degenerated and will disappear. Hence the vitelline vessels will also disappear. The allantoic diverticulum will disappear. You have learned it closes down. You will learn what it has done with uroreproductive system. And then we have only the mature umbilical cord containing what? The umbilical vessels, that means 2 arteries. Here you can see 2 arteries and 1 vein. This is the cross section of the umbilical cord in the lower figure, surrounded by extraembryonic mesoderm, which is also known as mesenchyme because it's primitive mesoderm. And in the umbilical cord, if you examine the umbilical cord after birth, it's like a jelly-like substance. Hopefully we'll see this specimen in the ward sometime. In the lab sometime. So here this is like a jelly-like substance known as Wharton's jelly, which forms a protective layer for the blood vessels, and the whole thing then is surrounded by amnion, as we have seen. Okay, and this is the normal length of the umbilical cord and the breadth, etc. Now if you take a histological section of the umbilical cord, see, it looks just like a face. The 2 eyes form the 2 umbilical arteries. You can make out how they are arteries because they are thick walled and more muscle in them, and lumen doesn't collapse, and there is a single umbilical vein because initially there were 2 but the one disappears. So usually it's the left umbilical vein that persists, the right disappears very early, hence we have this. So allantoic duct disappearing, and this is the Wharton's jelly or extraembryonic mesoderm, and this whole thing is surrounded by amnion. Okay, and that's the umbilical cord seen here. Now there are some terms to understand with the umbilical cord. So it has a certain length and a diameter we've seen. Now sometimes there are false knots. False knots means like this, that is just heaping up of the blood vessels. They are not actual knots like how we tie knots in a rope. Okay, because the blood vessels are quite tortuous like that, so that heaping up of these blood vessels is called false knot. But true knots, see, like the one shown in this figure, this is like a true knot that you would tie. This is in danger of obstructing the blood supply naturally, okay, to the fetus. Now sometimes the cord, if again too long or shorter cord, can cause problems. If it's too long, it can prolapse, or it can wrap itself around any part of the body, around the neck or any part. Again the cord can get obstructed, and this can affect fetal growth, movements, etc. A short cord may result in, the fetus is moving around in the amniotic fluid, and if the cord is short, it may pull on the placenta and causing hemorrhage and detach the placenta before, full term. Sometimes there is an agenesis of one of the umbilical arteries or any vessel, and this is usually associated with cardiac other vascular defects. Okay. So here we are. Note the normal anatomy now. That is the placenta developing. It has attached normally. It's higher up, and the amniochorion has fused. The decidua capsularis has degenerated. So this whole fetus has filled up the amniotic. Sorry, the uterine cavity, and is surrounded by the fused amniochorion. Now this is normal. So in this umbilical cord, what is happening? You can see the two umbilical arteries are shown in blue here because they are carrying deoxygenated blood and excreted metabolites, etc., to the placenta. And from the placenta, the single umbilical vein, which is a bigger diameter, is bringing oxygen and nutrients to the fetus. So that's the pathway, right? Now here what can happen? Here is an example of, say, a long cord which has wrapped itself around the fetus's neck. So what is the effect? Is it that the fetus is going to be choking with that cord? You feel as if the fetus may choke, right? But it's not the fetus that can choke, because the fetus anyway is not breathing through his lungs, but it's the cord that can be compressed by the neck between the chin and the chest. Can you understand that? And that's the result. If the cord is compressed, you can understand that it can result in fetal asphyxia or even death. Okay, so it's not the neck that's getting compressed, it's the reverse. The neck compresses the cord. Okay, and recall, we've already seen the normal attachment of cord when we were doing placenta. So it's eccentric towards one side, to the membranes, right? But sometimes if this umbilical cord is extremely attached only to the membranes, then it's called a filamentous attachment. It does not reach the placenta itself and only is attached to the amniotic membrane on the fetal side, and then it can be easily torn. The other kind is again a marginal attachment to the placenta, which is also not good. This is known as battledore placenta, like a badminton racket. Again, it can get torn, or obstruction of blood supply can occur. Okay. Now let's move on to an interesting aspect of twins. I'm sure some of you do have twins, so now you can know about yourselves. So you know that in the population, usually there are two kinds of twins. One is dizygotic or fraternal twins. The other is monozygotic twins. So what are these and how twinning happens, twin pregnancies happen? Let's learn. So these are the incidences worldwide, and there are many factors that are responsible for twin pregnancy in the family. So increasing maternal age, there's family history, that means genetic component is definitely there, etc. Now here what happens in dizygotic twin pregnancies is that if two secondary oocytes at ovulation are simultaneously released, they might be released from the same ovary or from different ovaries, it doesn't matter. So they are released simultaneously into the uterine tube. And you know that as it is, there are millions of sperms just waiting to fertilize oocytes if they find any. So here two secondary oocytes are fertilized by two different sperms at the same time. This will result naturally in formation of two independent zygotes, which will be in the uterine tubes, and then they will enter the uterine, then the same cleavage, etc., happens, and then they implant separately in the uterus. So each zygote, it's like two pregnancies but happening at the same time. So each zygote has its own placenta and membranes, right? They will have slightly different genetic constitutions. They will resemble each other just like any brother or sister or brother, and they can be of same or different sex. So they're not identical, but they can so they're not identical twins, right? That's why they're called fraternal twins, and this pregnancy is called dizygotic twin pregnancy. Now how can this happen? So here you can see, that means there are two zygotes and then two inner cell and outer cell masses, so two everything, and then finally the only thing is they occupy the same uterus. Sometimes they come so close together that if their two parts of their placentae may fuse, and even the amniochorions may fuse partially, and so if they come so close together, sometimes each twin may possess RBCs of two different cell lines, which is known as erythrocyte mosaicism, if some amount of RBCs have been exchanged due to intimate fusion of the placenta. It doesn't cause any problems. It's just that sometimes it may exist like that. Obviously, twin pregnancies have their own issues that both must get equal blood supply. We'll see what happens with clinical correlates about twin pregnancies later. Now coming to monozygotic twins, as the word monozygotic suggests, that means they have formed from one single zygote. So we'll see how this is possible. So here are pictures of identical twins. Usually they're more rarer, and they have, as you know, they have strong external or physical characteristics are the same, or a physical appearance. That means same eye, hair color, facial features, etc. What's important is they will have identical blood groups, whereas in the dizygotic they may have different blood groups. They are of the same sex, whereas in dizygotic twin pregnancies they may be of different sex. As you know, they can be same or different, but here they're always the same sex, as you can see in the pictures. And they will have almost the identical set of fingerprints. That's another important factor. Okay, and a similar genetic constitution, because they have developed from a single zygote. Now let's see the ways in which monozygotic twin pregnancies can occur. So as we said, it's only a single oocyte which is released, which matures into a single ovum fertilized by a single sperm. That's important. Now, but what happens once the fusion happens and the zygote is formed? This zygote, if it splits early into roughly at the 2-cell stage, it will result in two morulars, two blastocysts, so two inner cell masses, outer cell masses, etc. These will implant separately, but each will have, because it's early splitting, and the amniotic cavity, etc., develops in the inner cell mass. So and there are two blastocyst cavities, so each will have its own placenta, amniotic cavity, chorionic cavity, etc. And this arrangement resembles that of the dizygotic twins, but identical twins can be recognized by their common characteristics. Like, we will know that they are monozygotic twins because they will have the same blood group, same sex, same physical appearances, etc. Okay, same fingerprints. Now let's see what are the other ways in which monozygotic twin pregnancies can present, or arrangements can happen. Now, we have seen is when there is early splitting of the zygote at the 2-cell stage, and then this resembles dizygotic pregnancies. Now let's look at B. What's happening in B? So this 2-cell stage zygote became 4, etc., 8 morula, so that was single. The splitting happens at the early blastocyst stage. Now remember blastocyst, the structure of the blastocyst, so inner cell mass and outer cell mass. Now when that blastocyst forms, remember all these are pluripotent cells. If these inner cell mass splits up into two inner cell masses, you can understand that there will be two amniotic cavities, two bilaminar germ discs, hence two embryos. Okay, but they will have a common chorion and a common placenta, and that's what's happening in the subsequent figures, right? So separate amnion, because each amniotic cavity develops in each inner cell mass, right? And this is the most frequent mechanism. And then you can see in here they will have a common placenta because there is a common outer cell mass, but each of them will have their separate amniotic sacs. Okay, so common placenta and common chorion. Now the C is little more rare. Here what happens is there is late splitting of not even the inner cell mass, but late splitting of the bilaminar germ disc. Then there are two bilaminar germ discs, but one amniotic cavity. See, so they will have common placenta, common amniotic cavity, and common chorionic cavity, like that. So these are the three ways in which monozygotic twin pregnancies can present depending on when the splitting has occurred. One is A is early zygote, B is blastocyst stage, and C is late at the inner cell mass stage. Okay. Now you have heard of even still more multiple pregnancies like triplets, quadruplets, and quintuplets means 5. And people have already also heard of 7, 8 being delivered together. These are more naturally rarer and usually more common in mothers who have been using these given fertility drugs, because more gonadotrophins, more FSH, etc. to stimulate, so more follicles, more oocytes, and hence sperms are very happy to fertilize more oocytes, right? Obviously, the risks will be more because each baby would be smaller size, preterm labor or premature birth would be more common, twin defects, etc. So they would have to be very carefully monitored, and some may not continue to full term. Now, here usually there is family history, and as you know, the twin and multiple pregnancies will have a higher incidence of all these problems as we have seen. Now, let's briefly look at some of the defects associated with twin or multiple pregnancies. Now, any multiple pregnancy, be it twin or triplets or whatever, is always associated with increased risk to either one or both the fetuses. Now, one way this can happen is where one fetus gets all the blood supply and nourishment, so it grows more larger, and the other one you can see is shrinking and becomes very, very underdeveloped and will eventually die. So it becomes like a dehydrated fetus, which is known as fetus papyraceous. That's the term used there. Now, this usually can occur because of some twin transfusion syndrome, where where monozygotic twins who are sharing this chorion and placenta and everything, but there are some vascular anomalies in which can result in one fetus getting more blood supply, less the other one getting less. So that one will naturally become much smaller and may not even survive. Okay, usually it's some due to vascular imbalance and abnormal vascular blood flow. Okay, so just to be aware of this. Besides that, of course, there can be abnormalities of positioning, entanglement of the cord. So it's always like these twin pregnancies are better monitored, carefully monitored throughout pregnancy. Now, another thing that can happen, you've heard of these probably, they're called conjoined or Siamese twins. Conjoined means these twins end up being joined at some part or the other. Now, what is the embryological basis of this? Usually, remember, when there are two inner cell masses, two primitive streaks, and then each primitive streak will give rise to their own tissues, etc., right? But if this primitive streak does not separate completely, it separates only partial splitting of primitive streak and node at later stages, that means it has developed and it persists, like, it doesn't split. This results in, now remember, primitive streak are giving rise to what? It develops in the epiblast and gives rise to all the other tissues, right? So if that has not split properly, only partial splitting, then it can result in fusion of various body parts or organs and sharing of organs between twins. So, and there are different parts and names assigned to, depending on type of conjoined twins. Now, depending on the parts or organs shared, some conjoined twins can even survive and grow to adulthood. So we shall see examples of this. And some can be successfully surgically separated also after birth, provided there are no vital organs involved. So let's learn these phases. Now in this figure you can see this is almost a lot of fusion of different body parts, but the heads remain separate. So that is called dicephalic fetus. Obviously this is not very compatible with survival, but depends on the organs shared. Now, some of them are here. You can see if they share thorax, skin, or some organs. So it's called thoracopagus, pyo, pygopagus, where they are, joined at the back or spine. Now, to the extent to which we don't know, that will have to be investigated. Craniopagus, where their skulls are united, or here you can see again thoracopagus and even some abdomen. And here they were joined at the head like this, craniopagus, right? But see, they are survived like that. So here you can see dicephalus, even these survived. Probably, even if they're sharing some vital organs, as long as the vital organs are functioning normally, you see they can grow to a greater stage. And see how human adaptability, they are doing so many things together. It's like two, they will feel they are two, but they are sharing a common trunk and limbs, but the heads are separate. So these are compatible with survival. Now, the most famous case of the Siamese twins, as they were called, or conjoined twins, where they lived to in their 70s or 80s, I think. They were, in the 1800s, and they became famous, Chang and Eng, because people obviously thought they were weird, or some special people, so they went around the world giving shows like that, and they were joined at the thorax, and later on it was found that they they had shared a liver like that. They chose not to be separated. We don't know about the heart, so maybe the heart was separate. It's mostly at the abdomen. So, but they survived to adulthood. They got they had two wives and fathered 21 children. So not all congenital anomalies are fatal. Some are compatible with survival. So hope you found this interesting. And if you have any queries, of course you're welcome to post them. Now the ICA is going to be held as usual on Thursday, 4th November. So look out for guidelines and student distribution lists, which will be uploaded shortly. I mean, guidelines have already been uploaded. Look out for the student group distribution lists about the venues. Okay, thank you.